Welcome everyone to the third lecture of our fall lecture series. I'm Gina Norato, a statistician in the Office of Biostatistics with the Clinical Trials Unit in NINDS, and I'm going to be talking today about p-values. Please feel free to put any questions in the chat as they come up, and I can try to address them as I go. Okay, so to briefly go over the outline of what I want to talk about today, first I want to talk about what is a p-value. Next, I want to go over some common misinterpretations of what p-values are. Then I want to talk about how we can go about reporting and discussing p-values in the most accurate way possible. And finally, I want to talk about p-values in the literature and some of the controversies, I suppose, for lack of a better term, that have arisen over the past, at least decades about p-values. So firstly, what is a p-value? So in the strict statistical sense, there is a clear definition of a p-value, which is the probability of seeing data as extreme as or more extreme than the current data if the null hypothesis is true. In other words, if there is really no difference between the treatment and control groups under investigation, how often would we see data with at least our observed difference between the groups? Or how unlikely are our findings in a world where there is truly no difference between the groups? We can also sometimes think about p-values as like a continuous measure of compatibility with our data and the null hypothesis. So for example, a p-value of 0.59 could be considered high compatibility with the null, and a p-value of 0.10 is relatively low compatibility with the null hypothesis. So I think actually it's helpful to try to understand what p-values are through simulation and using that definition and simulation to help us get a better understanding of what these are. So let's imagine a scenario where we have two groups of patients, a treated and untreated group of patients, and we have taken systolic blood pressure from each group after treatment. And maybe we want to know, are the group's systolic blood pressures significantly different from each other? It's a very basic question. So we can just, for this example, I'm just creating some, fake data here. So in group A, our treated group, we have 8 people with a mean of 131.9. And group B is our untreated group, and there we have 12 people with a mean of 136.9. And so our estimated difference of A and B is -0.5 or -5.04. So we want to know something about the statistical significance of that difference as it occurs in this data. So we can actually, run a simple t-test between these groups and get a p-value, but we can also simulate a p-value. And again, I think this is sometimes a helpful way to understand how they work or where they kind of come from. So back to the definition of a p-value. Again, this is the probability of seeing data as extreme as or more extreme than the current data if the null hypothesis is true. And in this scenario, our null hypothesis is that there is no difference between the groups. And so we can simulate no difference between the groups by shuffling the data, or in another sense, kind of shuffling the group labels to pretend that there is no distinguishability between these two groups, and perhaps we can flip around their group membership and there's no real pattern in their group membership here. So again, this is our original data with the treated group in black and the untreated group in red. And so we can visually show what one potential shuffling of the data would look like if we pretend these groups really have no difference. We can shuffle them together. There's not really going to be any attachment to the labels here. So in a sample shuffle of number 1, what we would do in this simulation is take the mean of new group A and the mean of new group B. We store the calculated difference between those two new means, and then we would proceed to the next shuffling of the labels or of the data, depending on how you want to think about it. So again, second shuffle, we mix them up a different way. Now we have, mixed groups again between treated and untreated, and we take our new means. So we take the mean of our new group A, the mean of our new group B, we store that difference, that new calculated difference on the side, and then we proceed to the next shuffle. So after sufficiently many simulated shuffles, usually upwards of 1,000 or more, depending on your computational ability and how smooth you want these estimates to be, we can obtain kind of a null distribution of those mean differences. So every time we shuffle and calculate a new estimated mean difference, it informs this distribution here, where it will be centered around 0, and then we'll have some spread here. The red line on this graph is showing what our true, I shouldn't say true, what our observed mean difference was in our original analysis. And all of these points kind of show each calculated mean from each of our shuffled calculations. So every time we're shuffling, we're calculating that mean, and we can see them all plotting on here in this distribution. So for a two-sided test, we want to know how many tests in this null distribution are as or more extreme than our observed test. So again, here's our test, and we want to know kind of how many of those simulations give us something that's even more different from 0. And so in a two-sided way, we're talking about either more negative or more positive than the absolute value. And those are denoted in red here, those tests of interest in the extreme. So we can count the number of tests of, our 1,000 or, even more depending, but in this case, 1,000 simulations, which have absolute value that are greater than the absolute value of our observed mean difference, which is 5.04. And again, those are denoted in red here. And so for this simulation, this ends up being 1,316 simulations of 5,000, which gives us a simulated p-value of 0.263. And if we just do the usual thing, which is get a p-value from our two-sample t-test, we get a p-value of 0.283. And so we can see those are very close to each other. And so that hopefully gives some intuition as to what these things are and what that strict definition means. I feel like understanding it through simulation is a little bit more helpful, and seeing how this plays out is a little bit more intuitive than just thinking about the verbal definition. And in these settings, I think it's also somewhat useful to understand simulation too. So I think this does double duty here. And as you do more simulations, oh, in this case, I did 5,000, not 1,000. So as you do 10,000, 100,000, these numbers will get more close together. This estimate will get close to the p-value that you would get from your software. So along those lines, after kind of understanding what the p-value is defined as strictly, I think it's important to go over what a p-value is not. And we're going to talk about misinterpretation sort of throughout, but I think it's important here to highlight a couple just to get started. So here I have the definition reminder up at the top, which is the probability of seeing data as extreme as or more extreme than the current data if a null hypothesis is true. So again, if we assume there is no difference between the groups, how often would we see what we saw? So I have some ranked classifications of common misinterpretations, from somewhat less subtle to more subtle, or maybe more common misinterpretations. So that some people misinterpret p-values as being absolute proof for or against your hypotheses. And this is not true. They're really just probabilistic statements. And really, at most, all they can do is flag results as being unusual or provide a continuous level of corroborating evidence for the hypotheses. Next, misinterpretation is related to statements of reliability of the finding. So this is really in terms of whether you think that your finding would reproduce in another trial, or whether you think that your trial was good or that it's strong. So these are not good interpretations of a p-value. Small p-values are not more reliable than large p-values. all of them are subject to the same limitations or pitfalls within the context of your own study. And those have to be considered on the side. The next misinterpretation is the chance of making a type 1 error or making a mistake in your decision of the success of the trial or the significance of the trial. And this is not true because really this requires us knowing what the truth actually is to know whether or not we did make the mistake. And in reality, we'll talk briefly about type 1 error. There's not so much room for type 1 error discussions and all those things in this particular lecture, but in reality, that type 1 error false positive rate tends to be a lot higher. People argue that it's often higher than 0.05, our usual 0.05 cutoff anyway. So really thinking about it in those black and white terms is not that helpful. The next misinterpretation is defining it as the probability that the null hypothesis is true. And again, no, we already assume under that framework, we assume that the null hypothesis is true. We're assuming no difference between the groups. And p-values, strictly speaking, are not hypothesis probabilities, they're data probabilities, the likelihood of seeing data. And finally, this is some of these are pretty intricate, subtle definitions that sometimes take a while to think about. So I would encourage you to make note of some of these resources if you want to read more about these examples. But the last example is the probability that chance produced the results. So this is also a really common misinterpretation, saying that something's due to chance. This is partially because of the way our English phrasing of these things work, but to be strict, this is inaccurate. And so often we would say something like, if p-value is 0.08, there's an 8% probability that the results are due to chance. And again, this is sort of insinuating that we are able to know how correct we are with this particular result. And again, we're already assuming that chance is at work here. We're assuming that there is no difference between the groups and that anything that we see is coming from chance. And so, you know, when we leave it to this random process right here, we would see our observed results in 8% of studies performed. So that's like a more true representation of what that means. And we're going to come back to, we'll kind of jump back to some of these ideas too throughout the talk. So why is there so much confusion about the interpretation of p-values and why are there so many misinterpretations? And really it just comes down to that it's been perpetuated. In a little while, I'll talk about some research that's been done about this in particular, but in another way, we also just rely on them too much to interpret our data, one could argue. And we often want to contextualize our results in terms of p-values, and sometimes we are not as precise as we should be about the interpretation of those p-values or the results of the study in relation to the p-values. So again, I'll talk about kind of what to do about this and some issues and some considerations that come up. But again, I think we sort of, humans like black and white, sort of cut and dry answers, and in fact, p-values are really not that, and sometimes we hope that they are. And I think the other reason that there is confusion, and this is what some people suspect as well, that really what we intuitively want to know is actually the opposite interpretation of the p-value, or the converse interpretation. And I'm not going to go into Bayesian or frequentist statistics here so much, but in actuality, Bayesian statistics is usually more like what we would want to know. And what we really want to know is the probability of our null hypothesis being correct, that we have the observed data. But the frequentist interpretation, as we've talked about, is really the p-value is the probability of seeing our data, given that the null hypothesis is true. So there's sort of the flipped interpretation. But Bayesian statistics usually requires additional information about like the assumed likelihood of the hypotheses and all of those things. So, but this is why some people prefer Bayesian statistics, because of that interpretation of it. But again, they're sort of the complement of each other in that way. So we run our we get our data, we run our analysis, we get some p-values out, and we need to report them. So here I just wanted to put some, I wouldn't even call these conventions. This is kind of what I do. Every journal is going to be a little bit different. Some investigators may also have different preferences, but there are, this is one good way you could do this. So I will say you should always present the full p-value. You'll see sometimes in write-ups where people will write NS for not significant, or just say that the p-value is less than 0.05, is saying it was significant, but we're not going to report what the actual value is. I would say don't do this. You really want to report the full numeric number as properly rounded as you can get it, unless the p-value is prohibitively small. Obviously, we don't need to report to like 6 digits here. So you can say here in this example, you can just say less than 0.001 if it's very, very small. Also, if it's like medium smallness here, kind of the hundredth place area, you can round to 3 decimals to get a little bit more granular. And then if you're above 0.01, you should be rounding to 2 decimals. So that's just some rules of thumb, some examples here. So obviously 0.562, I would personally round to 0.56. We don't need 3 decimal places really. This one I would call less than 0.001. The 0.0079, I would round to 0.008, and 0.0145, I would round to 0.01. And again, part of the reason this is the biggest thing about actually just reporting a number here, as long as you have the space. Figures are a little bit different. You can kind of get away with just saying significant or not significant in a figure where you're trying to reduce visual bulk. But for text and for tables, it's important to not rely so much on that binary interpretation of the p-value and report the continuous p-value as you obtained it. The other thing I will say here with reporting is that p-values should really never be used alone, except maybe in an abstract if you're really hurting for space, but even then I wouldn't really recommend it. They should really always be paired with some other statistic, a mean and a confidence interval, a mean and standard deviation, medians, IQRs, things like that. Or some other statistic that you've obtained from your analysis, because that helps contextualize what that p-value is and where it's coming from, and it's forcing the reader to also think about the numbers in the scale that you have obtained. So you're talking about the mean of the actual the outcome measure, right? Instead of just talking about a p-value that's kind of without units, right? So being able to always relate it back to the data is very important. So also with reporting and in talking about your statistical plan, obviously type 1 error is going to be important here. This is the level at which we make those binary decisions of statistically significant and not statistically significant. And again, type 1 error is the probability of making a false positive claim. So claiming you have a significant result when you do not. The alpha level is hopefully set at the start of the trial, set a priori, and this is thought to be one way out of many, I suppose, to protect against incorrectly claiming significance. If you have done p-value adjustments for multiple comparisons, you should be reporting the adjusted values, obviously. And we could give like a whole talk on adjustments for multiple comparisons. I'm not going to go into that too much here because I want to focus to say specifically on p-values conceptually. But this may happen in scenarios where you've done repeated testing on related outcomes, or you're testing differences between groups on the same outcome, like you have 3 groups and you want to do pairwise comparisons on a single outcome. Or maybe you've run like a battery of 20 tests and you want to control for the fact that you've done many tests. That's another area where we would be thinking about adjusting type 1 error and being more strict about what we would call a significant versus a not significant p-value. So just to run through an example of how we would interpret a p-value and maybe how we would talk about it. So just to get through just an example, let's say we were testing a vaccine's effectiveness using a treatment and control group, and we've set our alpha level or significance level at 0.05. And again, our null hypothesis is going to be that there's no difference between the groups. And so we run our test, whatever test that we have specified that will be. It doesn't really matter here for this discussion. And we obtain a p-value of 0.03. So along with reporting all of the continuous data, the means, or whatever you want to report from that analysis, these are some interpretations that we can have given this p-value. One is that assuming that the vaccine has no effect, we would see the observed difference or more in 3% of studies. So that's one possible interpretation. Another interpretation would be that the data is unusual or not very compatible with the null hypothesis of no difference between the treatment or control groups. But again, the note here is that we really can't be sure that our study was a false positive or true positive. So we don't know. This is sort of where it's kind of down to probabilities in a way. And another interpretation would be at the specified significance level, the null hypothesis should be rejected, right? So this is talking about more of a binary interpretation of the p-value. So again, at this 0.05, we would reject the null and say that there is evidence of there being some difference between the groups. And I just want to highlight here that this does not mean that the null hypothesis is proved false or that the alternative hypothesis of some difference is proved true. So it's really important to avoid that sort of language. Again, this is sort of probabilities, and so nothing is proven. I will pause here to see if there's any questions before I jump into some of the controversies. I think that should take up the bulk of the rest of the talk. Okay, so all that being said, knowing kind of what p-values are and how we would like to talk about them, it's important to talk about the current literature, I guess, surrounding p-values and some of the discussions that are happening in the field and what we can kind of do to help this process. So p-values are in some ways considered controversial, and they're definitely under high scrutiny in, I guess, specifically the statistics community, but all communities that touch statistics, including like the medical literature, the psychological literature, biology, all of those fields that use these statistics and deal with p-values, all of those are touched by this. And I would say that this is due to several issues, some of which we've touched on, and I'll reiterate here. So one is this issue with the continual misinterpretation of what a p-value is and what it is for. The next is overinterpretation, so maybe understanding p-values but overstating, or just having too much confidence in your finding or trying to inflate what you found. Manipulation kind of goes along those lines, but I want to talk about this in more detail, about the ways in which we can inadvertently, in relying too much on p-values, sway our findings in the positive direction by making decisions throughout the research process that make a p-value more likely to be significant. And the last reason that I want to talk about for why they're under high scrutiny is just the relationship that p-values have to the publication process and how they're viewed in the literature specifically, how they're viewed by editors, how a significant finding really drives publication. And so I want to talk about that as well. So I see a question in the chat, but how would you make a regulatory decision, approve or disapprove the vaccine? Right, so one thing we'll talk about too towards the end is kind of what we should do. And one of the recommendations from the American Statistical Association is that p-values alone cannot tell you what to do. They are just another statistic. They're just another piece of information that are supposed to guide the decision-making process. Vaccines in particular are one of those things that there's a lot of pieces to that decision. There's safety information, there's efficacy information, there's funding, there's availability, there's resources, all of these things. So I think the feeling is that in particular, p-value should not be the one thing that sends a single trial through, bang, boom, we've approved the drug, like move it to phase 4, all that. So it's really about understanding your effect sizes, being able to reproduce a finding or replicate a finding in multiple studies, continuing to find the same effect, seeing signal across different sorts of environments, those sorts of things. So it's meant to be one piece of information and a holistic decision. I guess that's how I would respond to that. And I see another question. There are some discussions about p-value is outdated and should be replaced. Do you have any comments on that? Yeah, so we'll talk too about kind of what people are proposing, I guess, to do. I don't know what you mean by replaced. There are some proposals about whether they should be like omitted completely, but I don't know of any different statistic necessarily, or perhaps maybe we rely more on effect sizes or confidence intervals or other things like that. So there is discussion about what to do, obviously, and we'll get into some of that as well. And I would say it's not really that it's outdated. It's just if we are unable to fix people's over-reliance on it, how can we help? I think that's sort of the goal. How can we help people properly understand their data and not rely on a single value that their whole trial has to hinge on that? Okay, great. Thank you for your question. Okay, so starting off with misinterpretation, again, as we stated earlier, p-values are often misinterpreted. We went through a couple of examples. I do want to talk about this one paper in particular, which investigated some misinterpretations and how prevalent they actually were within the community. They administered a survey to students, teachers, and researchers, these 6 questions, and they asked them to say whether these were true or false definitions of, or what you can say in relation to a p-value. And so I'll just read them, I guess. So the first is that you have absolutely disproved the null hypothesis. That is, there is no difference between the population means. The second is you have found the probability of the null hypothesis being true. The third is you have absolutely proved your experimental hypothesis that there is a difference between the population means. The fourth is you can deduce the probability of the experimental hypothesis being true. The fifth is, if you decide to reject the null hypothesis, the probability that you are making the wrong decision. And finally, you have a reliable experimental finding in the sense that if, hypothetically, the experiment were repeated a great number of times, you would obtain a significant result on 99% of occasions. I'm not going to poll the crowd here. Spoilers are all false. And these were the findings. So this is their sort of figure 1 from their paper. This is their 3 groups from their study, and they pulled together information from a different study that asked similar questions in academic psychologists. And this is the percent of people who made at least one mistake in answering these questions. So all of the psychology students got it wrong. You could probably argue that that's because psychology students might be assuming that at least one of them has to be right. They're playing a little psychology game there. But again, these are very high percentages. Scientists not teaching methods, 89.7 of them made at least one mistake in this quiz, I guess. And methodology instructors, 80% of them made a mistake. And these are not huge samples, and this is a fairly old study. So I don't expect this to necessarily be true, but I think it's still an interesting look. And I'm sure that this was a time where people were talking about this as well. This is the original study was from 1986. So this is by no means a new discussion at all, but it continues to come up again and again. And they broke down the findings also by question. So we can kind of see where people were getting sort of tripped up. So again, this right column is the original study, and this is the current study. And so we can see where most people were kind of missing. These are the percents. Each of these numbers is the percent of different wrong answers. So a lot of people were getting these incorrect. So one is that the probability of the alternative hypothesis is found. and one is the probability of type 1 error. So these are really two really common misinterpretations, as we've talked about. And you can see even the instructors are kind of missing this. So, I mean, this isn't like alarmist or anything. Like this is more just a point to say that this is something that in some ways is really embedded in the field. And I think, again, I'll probably talk about this again at the end, but it's difficult to try to teach advanced statistical concepts to students who maybe get lost in the intricacies. And I think sometimes in an effort to explain, we try to simplify. And I think in the simplification is where we can have misinterpretation. So I think this is where some of the issues come from as well. And then they kind of just, they trickle through, and people use the same phrasing. You use the same phrasing for your papers, and it just becomes perpetuated. So I thought this was a pretty interesting paper. I don't know whether there's something I pulled a while ago. I don't know whether there's newer studies that have been done along these lines, and that would be something interesting to look at as well. The next thing I want to talk about is kind of overinterpretation. So along the same lines as misinterpretation, I think p-values can really be prone to overinterpretation. And I think this overinterpretation falls into a couple different types of categories. One is in relation to talking about binary statistical significance. So how can we properly talk about a result being significant or not? You know, a lot of people will, again, misinterpret, but then overinterpret p-values as really being proof that your study will replicate, or that your study is really strong, really well designed, all in relation to the p-value. And that is not the case. You have to make those conclusions based on, obviously, there may be other reasons why you think your study might replicate, or why your study is good or strong or trustworthy, right? But the p-value alone will not tell you that. Another thing is the media. And I think that in an effort to, again, this is going to come back to the literature thing I want to talk about at the end, but in order to make our research catchy and interesting, we sometimes overstate the findings, or we use a p-value to leverage discussion about other close significance results or things of that nature. And I think in an effort to sort of gloss over some of these intricacies, the statistical and scientific intricacies of your study, you allow other people to overinterpret your work as well. So it's really important when you write to err on the side of being conservative in your interpretation, because people tend to like skim and read what and kind of extract what they can quickly. And it's important to use language that acknowledges that the p-value is not going to be your end-all be-all for your study, and to view that as one part of your entire paper. Another way that we can think about overinterpretation is because p-values are directly related to sample size, we can often go down the wrong path in thinking that our result is strong or good just because we have a really large sample and are able to detect a small effect. Or conversely, we think that we have failed to find something just because our sample is small and we have a non-significant p-value. That would be one possible overinterpretation in relation to sample size. So some of you may know about XACD comics. They have a handful of statistics comics, but this is one that I feel like points to overinterpretation issues with p-values. We have our 0.05 cutoff here. We talk about these as being highly significant. Okay, these are significant. Okay, we're getting real close to 0.05. And this, 0.05 exactly. Oh no, we got to redo the calculations and see which side it's going to fall on, right? And here we call this on the edge of significance. And as we go up 0.07, 0.08, 0.09, highly suggestive, or significant at the 0.10 level, right? So this is obviously an exaggerated example, but we find ways to sort of talk about these things that make it sound better than it actually is. So I would, again, this is like a joke, but I would think carefully about how you're framing these things, and if you're giving proper weight to the effect sizes and all of the other aspects of your study that should be under consideration when you write up your research. And greater than 0.1, hey, look at this interesting subgroup analysis. So again, there's some truth to this. Obviously, we can think about a p-value as being a continuous level of evidence, as I said, but this highly suggestive or significant at a different level that we would maybe pre-specify normally, this is where you get kind of dangerous interpretations here. And then, as I said, this is kind of just an example of how you can have the same effect size, but different p-values as you have different n. So this is, again, just because they're proportional to each other, for lack of a better word. So imagine you ran a study with 100 people, you got an effect size of 0.25 and a p-value of 0.014, and you say, okay, great, wow, that was really good. Our study has a significant finding. Let's see if we can replicate it in a small additional group of people. So you run another study with 25 people, you get the same effect size, but it's not significant. And you're like, oh no, that's weird. That's now it's not, now we don't have a significant finding. So you pool them together, you have the same effect size and a significant p-value, you say, oh, okay, that's good. So this is where really your interpretation should be kind of the same, right? All that we're changing is the precision here as you increase n, and not really the effect size. So this is where, in like, I guess, epidemiology, for example, when you're working with very, very large n, p-values are completely meaningless because they're all going to be really, really, really small. So those discussions often hinge on effect sizes, or they should. And in other ways, in very, very small trials, a p-value may not be that useful because you're not going to have statistical power to talk about significant differences between groups. And in those cases, you really want to leverage your understanding of effect size. Is this a meaningful effect? Is this an effect we think we can elicit in other groups? All of those types of discussions are going to be helpful in making sure we're not relying too much on the p-value, because again, they are highly tied to the sample size. Okay, and then manipulation. Again, I don't like that word, but I didn't know what else to call it, so we'll talk about it. In the quest for statistical significance, in trying to find information about your research, investigators may intentionally, or more commonly unintentionally, alter the data, statistical analyses, or results of the study. And in all of those ways, you can kind of lose sight of the clinical effect or clinical meaningfulness, as I was talking about, in focusing too much on p-values. And this is a concept that was coined, this discussion of unintentional manipulation, I guess, was coined by this group. They're calling it researcher degrees of freedom. So this is something that will impact your research, that will skew what you're seeing. And the usual definition, the statistical definition, why this is kind of a funny phrase, or accurate phrase to call it, in statistics, the degrees of freedom is, kind of the number of independent pieces of information that went into calculating the estimate. And so this is to say that researcher degrees of freedom are the things that we do as researchers that impact the findings that we ultimately end up seeing, that we sometimes aren't even aware of. Kind of like, I don't want to say like butterfly effect, but there are many, 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 many decisions that go into the design of a trial, and all of those things will impact the findings that you obtain. Not like altering numbers, right, in your spreadsheet, like changing a number from an 8 to a 10 to make it look better, but we're talking about all the things that made your research at the end of the day. What if we had decided to make a different decision? We would have probably gotten a different result. And being able to acknowledge that branching of possible decisions that ultimately get us to where we end up. And so in their paper, I'll just read here how they talk about this, because I think it's very pointed. So they say, In the course of collecting and analyzing data, researchers have many decisions to make. Should more data be collected? Should some observations be excluded? Which conditions should be combined and which ones compared? Which control variables should be considered? Should specific measures be combined or transformed, or both? It is rare and sometimes impractical for researchers to make all of these decisions beforehand. Rather, it is common and accepted practice for researchers to explore various analytic alternatives, to search for a combination that yields statistical significance, and then to report only what worked. This exploratory behavior is not the byproduct of malicious intent, but rather the result of two factors: ambiguity in how best to make these decisions, and the researcher's desire to find a statistically significant result. And so I think many of you may be familiar with this concept, in that, you set out to do one thing perhaps, and we have to change what we're doing, and or maybe some type of sampling wasn't working, or we had to ask patients a different type of question to elicit the information that we wanted. All of these things have the potential to change your research, are all things that also have the potential to influence your p-value at the end of the day. Like, that's where this whole thing kind of ultimately ends up. And by trying to think about this p-value as this clean thing, and we can make a binary decision with a very clean type 1 error that we said at the beginning, it sort of glosses over what they're talking about here, which is all of these other things that have the potential to impact it. We can do our best with, like multiple comparisons correction or all these other things, but if we can't report and acknowledge all of the things that we tried that didn't work, or observations that we excluded that we didn't think to tell the field about, that all will be relevant for contextualizing your research, all of that is going to be important for helping people to understand the strength of your study and contextualize your p-values as well. So I see another comment, which I'll just read. It is interesting that effect size is not as popular as p-value. We end up searching / guessing the effect size on the papers, which is often being a very small effect size. Yeah, and again, I think it's kind of just, some of it's journal dependent, and you get used to writing certain ways. And if it's honest, some of that is honestly going to be on the editor and the reviewers to really stand up and ask for these things to be presented in a different way. And actually, that's a good lead in to the next section, is that you can't, it's one thing to understand what these things are and to want to write your paper in a good way, and to also want your paper to be published. And especially because null findings or, weak findings, perhaps in the p-value sense, they're often difficult to publish. Journals aren't going to make as much space for those papers. Not in all cases, obviously there are some very high profile null papers out there, but it can be difficult to want to publish those things. But in fact, all of that, if you're going to be responsible about it, all of that furthers research. And so we have to really try in our writing to present well what we do and why it's important, even if it is null. And instead of trying to make something sound, catchier or high impact, trying to do your data better by being true to the data versus trying to inflate what you're finding. And so, yeah, I think what you're talking about here is very relevant to the publication issue, and the pressure really that investigators feel on being able to get that significant less than 0.05 primary, result here for your paper. And it's just, I think I think people are starting to see it more, that it's kind of like a toxic way to be publishing, to really just be encouraging people to do this, to make it really hinge on that publication, instead of considering what would be best for the literature or the scientific community. Obviously, we're not going to publish every null finding or every weak finding if we think that it was poorly designed or there were some other issues here. But if we have a really strong, good study and we have a surprising null result, a lot of those can be useful. And so I think here this is another issue that people have, is we can make these leaps and these improvements in how we talk about p-values, but ultimately if an editor or a reviewer doesn't understand or turns up their nose or won't publish your research because it doesn't sound as good, right, then it kind of makes you want to just go back to how you were doing it before, right? So that's like the pessimistic view, but this is something that's discussed in the field, is that it's kind of a multi-level problem, that there needs to be change from a lot of different areas in order for something better to be done here, and it's really not just going to come from one area. And so there are some proposed solutions. I don't think that this is an exhaustive list. There's like a huge literature on this, but I think this will cover the most high-profile ones. So one proposed solution for dealing with p-values and the controversies that come up with it is to only use confidence intervals and not p-values. So there are obviously some benefits to this because you're just not using a p-value anymore, but ultimately you can still extract binary statistical significance from a confidence interval if you understand what confidence intervals are. So you could still use it to say this is significant or not. But in fact, there are some benefits to this because it does, as I alluded to earlier, it does force you to stay in the scale of your variable. So when we're talking about confidence intervals, we're talking about kind of the range you're talking about in the original scale of your data, not p-values, which are really just different statistics. So it keeps people focused more on your effect size when you talk about confidence intervals. So pros and cons to this approach. It's also been proposed to make the threshold more strict, which has been proposed in one instance to be 0.005. This probably depends on the field. I'm not going to poo-poo this too much. I think this was specific for certain like high throughput data type applications where we're working with very large sample, and probably 0.05 is much too high in those scenarios. But this really doesn't. This cutoff value, I think, is also problematic and can really just lend a lot of misinterpretation or over-reliance on the p-value when we're talking about trying to put it in a binary framework. And so this probably solves some issues, and we're going to get less false positives, but we may also be missing real effects when we really are strict like this. And so this may or may not be a helpful approach. And in the most extreme, there is actually one journal. It seems that this is still the case today, but in 2015, Basic and Applied Social Psychology banned the use of the null hypothesis significance testing procedures, which means that all of the research that is reported there only focuses on strong descriptive statistics, effect sizes, and they encourage you to have a large sample size. So those things are hopefully more reliable because it's a larger sample rather than being skewed too much because it's a small sample. And so they were just like, well, don't run tests. We're just going to talk about descriptives here, or as much as is possible, talk about like effect sizes and differences in that way. So that would be like obviously the far end of the spectrum. So there really isn't one good approach right now. And I think, again, like I said, it also depends on the area that you are publishing in. They're all going to have slightly different conventions for these things, and each area is going to have its own issues or limitations for talking about type 1 error too. Like I said, here in some scenarios, we're better suited to cut this really low, but in other fields where we don't want to miss the finding, this isn't a really good suggestion here. Other sorts of recommendations that have been proposed, so from the Simmons paper that we just talked about, they make a couple recommendations. A lot of this is open reporting. So just being open with, aside from what to do with the p-value, focus on everything that led up to that as a discussion point for how all of the things that went into the design of and collection of the data. So they say you should decide the rule for terminating data collection before it begins and report this rule in the article. They give this sort of threshold or guideline of 20 observations per cell. or provide a compelling cost of data collection justification, right? List all the variables that you collected, report all experimental conditions, including failed manipulations, right? These are all things that can really determine the outcome of your study. And if you omit these, people may have too much confidence, in layman's terms, in your p-value, when in reality, maybe there were a lot of other things that you tried that didn't end up working. Report all analyses with and without excluded observations, so sort of sensitivity analyses here, and with and without included covariates. So again, just seeing, and again, going back to publication, you say, well, I don't have space to do all this stuff. I agree. There really isn't good space to do all of this, but it's a good thing to shoot for, and it's a good thing to keep in mind, and really focusing on those key things, maybe especially failed manipulations, being able to report those things as part of your paper is all really important. And specifically from the American Statistical Association in 2016 about this whole set of topics. So a lot of these recommendations that they put together on this discussion all harken back to things that we've already talked about, but this is specific from them. This is kind of where they stand on this right now, although this is now 6 years old still. They say p-values can indicate how incompatible the data are with the specified statistical model, right? So how we talk about them being a continuous measure of evidence, right? P-values do not measure the probability that the studied hypothesis is true, or the probability that the data were produced by random chance alone. So we've talked about those misinterpretations. And this goes back to the question we had earlier, scientific conclusions and business or policy decisions should not be based only on whether a p-value passes a specific threshold. So that goes to everything we talked about earlier in terms of really that one statistic is not going to help you decide how to move forward. It's information, just as much information as the mean, just as much information as the number of people you've had. It's just information. And putting too much weight on this is what's really damaging. Proper inference requires full reporting and transparency. So again, that goes back to what we just talked about with the Simmons paper, really being able to describe fully what you did and all potential issues you ran into, or how you excluded groups, how you grouped variables together to do analyses, all of those things, all of that is going to impact your p-value. P-value or statistical significance does not measure the size of an effect or the importance of a result, right? So size of an effect, as we talked about too, you can have the same effect size, but with different p-values if you have a different sample size. And the importance of a result, obviously, it doesn't tell you whether the result is good or not. It just tells you perhaps how likely it is in layman's terms, right? So it's not telling you, a p-value of 0.0001 is not more important than one of 0.9, right? Maybe that seems obvious, but it's just an important reminder. And finally, by itself, a p-value does not provide a good measure of evidence regarding a model or hypothesis, because really this is like again, one piece of information. This is sort of it's supposed to be kind of a holistic decision about what to do with your research. And I think even in, clinical trials, you can't just get something approved with one clinical trial. It's a process. You have to do replications. You have to show that something replicates in a different population or at a different center. You don't just get your drug pushed through with one p-value 0.03, right? So and I think that's also an important thing to remember, that that's really not necessarily how it works. It'll get you published, right? So that's where we want to be careful. But in fact, we tend to not just use that one thing to push through to approval. And so I guess maybe to summarize the recommendations, and some of these are, I guess, my personal recommendations, but again, there's like a lot going on here. So I think really in terms of just overall publication, just be transparent, report as much as you can about what you did, tried, and what you found. And all of that is to kind of just de-emphasize the statistical significance. And when you do use it, when you do talk about binary statistical significance, it should be in relation to a comparison that you've powered on. Otherwise, we don't really have any good interpretation here. And be sure that you're interpreting it properly, and be sure that you're thinking about those common misinterpretations. And be aware of when you are making these research or analysis decisions, those research or degrees of freedom, and how you might be inadvertently, obviously, biasing your results, or how you might have made a different decision and obtained a different result, and whether or not that's relevant for your findings. And again, I think that including like this publication issue, it really is just there needs to be like a larger cultural shift. It's kind of like a strong phrase to use, but there's so many pieces, there's so many moving parts to publication and to research. All those people kind of need to be on the same page when we are talking about revamping the use of p-values in whatever way that people think it's best to do that, and making space in journals for null findings, making space in journals for supplemental tables and methods where we can talk about what we did that didn't work, making sure that educators are informed about these common trip-ups and how to properly introduce even introductory statistics in a way that's accurate. And, in researchers not trying to think carefully about these things and reach out for help when you need it as well. It is not just on the researchers to write these things up properly. It's the statisticians, it's everybody. And then also, the funding groups, those are people who also have a hand in this too, being able to understand all of these issues. So that's where everything is right now. Again, there's a huge literature on this, and actually the ASA statement has a lot of sub-statements that were published alongside of it, all of which are extremely interesting. So if any of this sparks any interest for you, I haven't read all of them, but a lot of them should be accessible to the non-statistician, or at least large pieces of it, because this is an issue that applies to everybody. So if you're at all interested, I can also provide resources for that. But that just leaves us a couple minutes. Any questions or comments before we fully wrap up? So I see a question, what is p-hacking? So I would say it's most related to the idea of this researcher degrees of freedom, where p-hacking can be intentional or unintentional, right? So it's just a way of saying, so you've done something to put yourself in a scenario where you're more likely to get a positive result. And so it could be in a malicious way, and it could also just be because you've made a lot of upstream decisions that led you to your ultimate finding, and maybe you cut out a lot of outliers that you shouldn't have cut out, or you didn't like that one test that you ran, but this other test got a significant result, so you report that one and not the other one that wasn't significant. So that's what p-hacking is. I think it's like a catchy, funny term. I don't think that it really, I like the idea of researcher degrees of freedom better, because I think it kind of gets at the issue, and it removes a lot of the idea that some people are doing this maliciously all the time. And I think you think, oh, I'm not p-hacking, but I think actually it's a lot more subtle than that. And so we really have to be careful about how we're thinking about our data and what we're choosing to analyze. That's a good question. Yeah, it's definitely a researcher degrees of freedom. It's definitely more inclusive to unintentional manipulation of the data or unintentional influence for the final result. But p-hacking is more catchy. All right, well, my email is at the beginning. Many of you know how to contact me. Thank you all for joining in today and listening. And please contact me also if you have any more questions.